reliable their testing would be on a very complex sample like Flask 1029, we and they interpret the results to mean that, in fact, a sample of that sort, a flask of that sort, is potentially heterogeneous. It's heterogeneous in its composition and even its spatial organization, so that every time one dips into something so complex, you aren't necessarily pulling out the exact identical set of material. And that's a very important issue with testing of biological samples. The results also indicate potentially that each of these molecular assays is not going to provide a consistent result every single time it's performed, regardless of the context in which it's used.
Next question.
Scott Shane, New York Times again. When your review was announced, there was some criticism of the notion of reviewing only the science and not the sort of detective work involved in the investigation. I wonder if, looking back, you think that was a wise decision. I realize it was dictated by your customer, so to speak, the FBI. But from a public interest standpoint, with a case in which the person identified by the FBI has killed himself and isn't going to go on trial, in retrospect, would there have been a case for either enlarging the panel with other expertise, but in some way trying to review the entire investigation as opposed to just the science?
I think that one should strongly consider being able to put together interdisciplinary panels that would be able to do a review such as you recommend. I think for the composition of our panel and for the sheer magnitude of work just to review the scientific side of this investigation, this was an opportunity to provide a really in-depth look at how science is performed in the context of a national emergency and in the context of a law enforcement investigation with many experts being worked with across the country. And so I believe we provide an excellent service to the community in taking a look at how that is done and being able to prepare in the event of a future attack. I do think, and I'm a member of the Committee on Science, Technology, and Law at the Academies, science and technology and the law do come together more and more in our world, and there are important aspects of science entering into the courtroom that committee grapples with and other studies grapple with, and I do think that it could be fruitful in a similar scenario in the future to have such a study.
Just to add, this point also emphasizes the importance of our second recommendation, which is the value of clear, concise, but accurate communication of the realistic expectations from the use of science alone, as well as science together with the other components of an investigation like this. And that's going to be a challenge for us all in trying to do this even better the next time.
I have another question emailed in from a journalist watching online. Dave Altamari from the Hartford Courant asks, are there tests that you would recommend be done now comparing the anthrax and the attack letters to the anthrax in RMR 1029 that could provide a clearer picture as to whether the attack anthrax did indeed come from that batch?
Since this case really rests on a complex interplay between the science and the law enforcement investigation, we're not in a position to say whether additional science would be needed or beneficial. It depends upon the relative weighting between the science and the law enforcement sides of the investigation. We do in our report point out that today there are technologies available that can provide additional information about genetic signatures, and there are opportunities to look at the genetic mutations in a more detailed way than they were at the time. There are also opportunities to consider environmental samples that were not tested on a molecular level. But again, we do not make a recommendation whether or not those should be pursued. They really have to be weighed as to their benefit relative to the overall investigation.
Next question.
Hi, I'm Megan Eckstein with the Frederick News Post. I was just wondering if during the course of your studies you were able to have an interaction with scientists out at USAMRIID and how that may or may not have affected the conclusions you came to.
As I think you know, we did have one of the scientists from USAMRIID who was involved in work on Bacillus anthracis come and speak before our committee and answer questions on technical and scientific matters concerning the production of B. anthracis spores. We view that solely in the context of trying to understand the science better, both the science that's possible and the science that could be done under a variety of different circumstances. We tried very hard to dissociate who this person was exactly and where they worked from the information that they were sharing with us.
Question?
I just wanted to ask a question to clarify something it's in the document. As I understand what you're saying here on page 108, you are suggesting that some of these morphotypes, if not all of them, were present in the material that was cultured in the fermenter at Dugway. And if I understand that correctly, that would seem to raise a significant question about the cornerstone of this case, which is that the attack spores came from and could only have come from RMR 1029. Since you're finding these morphotypes before it ever arrived at Fort Detrick, it would seem to me, if I understand what you're saying correctly, that in fact you cannot say definitively that the attack spores came from RMR 1029.
There are a couple of different points embedded in your question. I think they're important and worth discussing briefly. First, we know from the scientific literature that mutations will arise in cells of B. anthracis when they are grown, and that particular mutations will be selected, will become enriched during certain kinds of procedures, and in particular during the course of batch production, large-scale production of spores. We know that mutations of this sort, as those found in the letters, do arise during batch production of B. anthracis. When we looked at the limited information given to us about practices and procedures at Dugway, we found that their procedures might well, in fact, allow for not only the propagation of these mutations, but the possibility that mutations might also arise during their processes and procedures. The second point, though, is the relationship of the Dugway work to the origins of 1029, and that relationship